In our last video for Chapter 4, we're going to finish our exploration of prokaryotic diversity by looking at two important groups, the deeply branching bacteria and the Archaea. We'll see that section 4.5 is pretty short and covers the deeply branching bacteria, and 4.6 will talk about Archaea. Deeply branching bacteria represent some of the earliest lineages to evolve after the last universal common ancestor, or LUCA. They often thrive in hot, extreme conditions that resemble early Earth, like hot conditions. So microbes that like hot conditions are thermophiles. Extremely hot conditions are hyperthermophiles. And studying these bacteria helps scientists understand what ancient microbial life might have been like billions of years ago. Our first example is the Pseudothermus paucivorans, which was discovered in sewage sludge in 1988. This is a thermophile growing best at about 58 degrees Celsius, so pretty warm. And it's considered one of the deepest branching bacteria, making it one of the closest known relatives to LUCA. So we can see it down here. And it's one of the first non-LUCA forms of life, produced by evolution probably around 3.5 billion years ago. Our next group of deeply branching bacteria include the class Aquifici or Aquifici. These include hyperthermophiles like Aquifex, so this is a genus name. These grow in hot springs and thermal vents at temperatures that are extremely hot, greater than 90 degrees Celsius, some even at about 138 degrees Celsius. And they're interesting because they use inorganic substances as their nutrients. And this is in contrast to, like us, humans, we use organic substances like glucose, carbon-based nutrients. These bacteria use inorganic substances because they have the ability to reduce oxygen or nitrogen gas for energy. So reduce refers to redox reactions that you learned about in chemistry, where reduction refers to the gaining of electrons. Finally, these bacteria are also very highly resistant to UV as well as ionizing radiation. Our next group of deeply branching bacteria are in the class Thermatidae or Thermatidae. These are anaerobic bacteria that are mostly hyperthermophiles. They like very hot temperatures, although some are mesophiles, which like more moderate temperatures, similar to the body temperature of us, 37 degrees Celsius or close. They're unique in that they have a unique sheath-like toga that gives them a pretty unique appearance. And their thin peptidoglycan wall also contains different amino acids, pretty unique amino acids that you don't usually see in other bacteria. A specific example here, Thermatidae maritima lives near ocean vents, and you can see it's a hyperthermophile. And these bacteria can also produce molecular hydrogen, which gives them potential industrial applications. Our last example in the deeply branching bacteria are the species Deinococcus radiodurans. So this is the genus name, and this is the specific epithet for this bacterium. This bacterium is sometimes nicknamed Conan the bacterium because of its extreme resilience. So it's a play on words from Conan the Barbarian, which is a fictional character you might have heard of before. Conan the Barbarian is known for his strength, combat skills, and survival instinct, and you might have seen him before in comic books or novels or different films. There's even a movie, Conan the Barbarian, that was played by Arnold Schwarzenegger in the 1980s. Going back to the bacteria, Deinococcus can survive radiation doses that would kill all other known bacteria, so thousands of times greater than what would kill a human. And they could even survive the intense radiation from like an atomic bomb explosion. So it wouldn't survive probably the heat or force of the blast itself, but the radiation after effect. Because it can survive these harshest conditions on Earth, we put them in the polyextremophile category, where they can survive many different types of extreme conditions: heat, drought, vacuum, acidity, and radiation. And the reason they can is because of their unique DNA repair mechanisms. So for some reason, even when their DNA is completely or almost completely damaged, they still have enzymes that have the ability to repair that damaged DNA. All right, let's recap 4.5, which is a pretty short section. Looking at the first bullet point, unique features include that they're among the earliest evolutionary linkages or lineages of bacteria. This branch was close to the last universal common ancestor, or LUCA. They're often thermophiles or hyperthermophiles, living in pretty extreme heat conditions similar to early Earth. Some of them were polyextremophiles, where they could survive radiation, drought, or acidity, and they provided important insights into ancient environments and the evolution of life. Looking at the second bullet point, give examples of significant deeply branching bacteria. We had several. So we had Acetothermus paucivorans, which was a thermophile and considered one of the closest relatives to LUCA. We had aquifex, so these could be hyperthermophiles that we could find in hot springs or thermal vents and are resistant to radiation. We had Thermotoga maritima, which had that sheath-like toga and grew near ocean vents. These also can produce hydrogen. And Deinococcus radiodurans, the cone in the bacterium, which is a polyextremophile and can survive extreme radiation because of their unique DNA repair mechanisms. In our last section, 4.6, we're going to switch gears and no longer talk about bacteria, but look at the domain Archaea. So remember, bacteria, Archaea, and Eukarya are our three domains. Archaea are still prokaryotes, but they're different from bacteria. Archaea form their own domain, separate from bacteria and eukaryotes, and they are more similar to eukaryotes than bacteria are. Archaea, like bacteria, are also unicellular, and they're also found in many different types of habitats, including extreme environments. Unique features of Archaea include unique lipid linkages in their cell membrane. So you don't have to know the details like ether links, but just know they're different in their cell membrane composition. They also do not have peptidoglycan in their cell walls. Instead, some Archaea have a structure known as pseudopeptidoglycan. Their genomes are larger and more complex than bacterial genomes, and they're more similar to eukaryotes than bacteria are. And then importantly, they're not known to cause any diseases in humans yet. Our first phylum is Archaeota, which is a type of Archaean, and these are extremely diverse. They're all aquatic, and you see many of them in the oceans. Many of them are hyperthermophiles as well as acidophiles. One specific example, Cephalobus, is a thermophile that survives high temperatures, 70 to 80 degrees Celsius, as well as an acidophile that survives pretty low pH. This bacterium can oxidize sulfur, and it can also produce aphetins, which are thermostable proteins that can actually bind to and neutralize different antigens like toxins or infectious agents. So right now, scientists are trying to modify these aphetins to use them as therapeutic drugs, such as inhibitors for the virus that causes COVID or other microbes. Another example here is Thermoproteus. This is a bacterium that grows well at 85 degrees Celsius. It's a strict anaerobe, and it also can reduce sulfur or even hydrogen while fixing carbon. We see a lot of these in aquatic environments like the ocean. All right, so we just saw the phylum Archaeota. These are aquatic, abundant in oceans, extremophiles, so they thrive in very hot conditions like 70 to 80 degrees Celsius, and acidic environments, pH 2 to 3. We just talked about Cephalobus, which is a thermoacidophile, and Thermoproteus, which is a strictly anaerobic hyperthermophile. Our next phylum are the Archaeota, and these include methanogens and halophiles. Methanogens can reduce carbon dioxide to form methane, or CH4. They're found in hot springs, glacial ice, and even the guts of animals, including humans. We also have halophiles in this group, and halophiles are salt-loving Archaea. They require nearly saturated salt concentrations, so super salty bodies of water, like the Dead Sea or the Salt Lakes. They can actually perform photosynthesis, and they have a pigment called bacteriorhodopsin that they use to harvest light energy, and it also gives them a distinct purple color. The most popular or well-known halophile is probably this one, Halobacterium salinarum. I know it sounds like bacterium, but remember that this is a type of archaean. This is Archaea, and this is possibly one of the oldest living organisms. Our book provides us with a picture of these salt ponds where halobacteria are often found, and Halobacterium salinarum, remember, has that bacteriorhodopsin, which gives them that purple color. And I can kind of see the purple everywhere. So these, again, Halobacterium salinarum might be one of the oldest living organisms because they've survived extreme hypersaline environments for billions of years and they're highly adapted. So this does not mean, though, that they're older than deeply branching bacteria. So bacteria came first, so those are the oldest. It just means that these Archaea, especially Halobacteria, have a very ancient lineage within the Archaea domain. And I wanted to include salt ponds from our area. So these are the San Francisco Bay salt ponds. These are salt evaporation ponds that are used to extract salt from seawater. And I can see there are different distinct colors here and there. Many of these contain halobacteria. For our halobacteria checkpoint, remember that these are found in hypersaline environments, so very salty environments requiring nearly saturated salt levels. Examples include the Dead Sea, the Great Salt Lake, and Utah salt ponds, evaporating seawater pools, and their reddish purple color comes from that pigment, bacteriorhodopsin, that they use for photosynthesis. Right, so key takeaways from these last two sections, 4.5 and 4.6. Deeply branching bacteria were the ancient lineages closest to Leuca, and they often thrived in extreme heat and radiation. For Archaea, they are unique microorganisms that have unusual cell structures and metabolisms, and many of these are extremophiles. Of the Archaea, we have two major groups, the Archaeota, which include thermophiles and acidophiles, and we have the second phylum, Archaeota, which include methanogens that produce methane, and halophiles that like those salty environments. So all of these groups really expand our understanding of the diversity of life and the conditions that might have shaped early evolution. And let's recap 4.6, Archaea. So for the first one, unique features of each category, we have the Archaeota. These are mostly hyperthermophiles and acidophiles that thrive in hot, acidic, aquatic environments, like Cephalobus, which was a thermoacidophile, and Thermoproteus, which was a strictly anaerobic, sulfur-reducing archaean. We also have our phylum Archaeota, which included methanogens. Remember, methanogens can reduce carbon dioxide and produce methane. And these are found in hot springs, ice, and even guts of animals. They also included halophiles. So within the Archaeota phylum, we had halophiles that require high salt concentrations, and they used that pigment bacteriorhodopsin for photosynthesis. So the most common example here were the Halobacterium salinarum. For the second one, explain why Archaea might not be associated with human microbiomes or pathology. So Archaea have unique cell structures. Remember, in their lipid membrane, they actually have ether linkages, so different linkages than other prokaryotes like bacteria. They also have no peptidoglycan. And they also have unique specialized metabolisms that are adapted to extreme environments. So they're not known to cause diseases in humans, probably because they don't compete well with other organisms in that same environment. And they also don't have any virulence factors that we know of so far. So virulence factors are factors that allow microbes to cause disease, to invade our immune system, and are basically what we usually use to fight off microbes. They can't evade them. There are some Archaea present in human microbiomes, like we do have methanogens in the gut, but they're not linked to disease. For the last one, give common examples of Archaea. So there are several. We talked about methanogens. So we have methanogens in the human gut and also in the guts of ruminants, like cows. Halophiles, so we saw Halobacterium salinarum in the Dead Sea and different salt evaporation flats, or these ponds. We have thermoacidophiles like cephalobus in hot acidic springs, and hyperthermophiles like Thermoproteus near volcanic vents. That takes us to the end of chapter 4. So finally, we're done with prokaryotes. In chapter 5, we're going to look at eukaryotes.